Bismillahirrahmanirrahim and Assalamu alaikum. We're here with another video for the topic gluconeogenesis. Okay, so gluconeogenesis, what does it mean? The first part, gluco, is for glucose. Neo means a new form, a renewed form, and genesis means to produce, to make, right? So gluconeogenesis essentially means that we're producing glucose from non-carbohydrate precursors, especially when the glycogen stores are depleted in our body and there is also no consumption of carbohydrates, right? We're not intaking carbohydrates and neither is there any storage of glycogen. This occurs during fast, right? When we keep a fast, gluconeogenesis is occurring. In an overnight fast, 90% of your gluconeogenesis occurs in the liver, while 10% occurs in the kidneys. For prolonged fasting, up to 40% could occur in the kidneys, right? So, what is it all about? It's actually almost a total reversal of the steps of glycolysis, because glycolysis was breakdown of glucose to ultimately yield ATP, but here we're making glucose to increase your glucose levels, right? Because the glucose levels have dropped, so we're making glucose. So we have to go back in the reverse direction. So what happens is that there are basically three main precursors. You can get it from triacylglycerols, the TAGs, which are your fats, your adipose, right? When you're not eating enough food, you say you're dieting, but your fat stores are depleted, right? That's why you lose weight. Triacylglycerols, what happens is that they're hydrolyzed to form glycerol and fatty acids, which is what TAG is made of. Glycerol amongst them is phosphorylated to glycerol 3-phosphate by glycerol kinase. Glycerol 3-phosphate then becomes DHAP, which is an intermediate of glycolysis by the enzyme glycerol 3-phosphate dehydrogenase, right? So DHAP then can go back to produce glucose, right? Okay, so another candidate for gluconeogenesis is lactate, which is constantly being produced under anaerobic conditions. That lactate, via a cycle known as the lactic acid cycle, also called the Cori cycle, enters the liver where it is oxidized to pyruvate and then ultimately yields glucose, and we'll learn how. So what happens is that lactate, you see, only forms under anaerobic conditions, right? And under anaerobic conditions, we're very much depleted of energy, right? Our energy stores are down because we've exercised our muscle to a limit where oxygen supply is inadequate, and so would be the energy, right? So Allah has made this that lactate contains a lot of energy potential, and it can be reoxidized to pyruvate to produce glucose, right? So it is a backup for us. Another compound that can form glucose are the amino acids, right? Your proteins can also be hydrolyzed to produce amino acids, which then produce alpha keto acids. Remember, alpha keto acids do not have nitrogen. Please do not confuse them with that. They're just alpha keto acids, alright? If that makes sense. Alpha keto acids such as pyruvate is an alpha keto acid, or alpha ketoglutarate. Alpha ketoglutarate, as I just mentioned in the TCH cycle, in the video on the TCH cycle, it is an intermediate in the TCH cycle, so it can ultimately yield oxaloacetate, which is converted to phosphoenol pyruvate, I'll just explain. And similarly, pyruvate can also be converted to oxaloacetate. Now, how does that occur? We'll see. Alright, so we were done with triacylglycerols. Now comes lactate and amino acids. Lactate, as I just explained, through a cycle called the lactic acid cycle, is oxidized in the liver to pyruvate. Now, pyruvate here is trapped in a way that, here I've drawn the last step of glycolysis. I did not mention all the steps because I didn't have space. With this last process is very important because pyruvate cannot be converted back to phosphoenol pyruvate because this was an irreversible reaction. The conversion of phosphoenol pyruvate to pyruvate can occur, but the reverse process cannot occur, right? So pyruvate has to offer an alternate route, right? And that alternate route is in the form of oxaloacetate. Pyruvate is converted to oxaloacetate by an enzyme called pyruvate carboxylase, right? Because pyruvate itself is a 3-carbon compound, oxaloacetate is a 4-carbon compound. So, oxaloacetate now has two routes, right? It can either go into the direction of the phosphoenol pyruvate, it can convert directly to phosphoenol pyruvate, or it can go to TCA cycle. Guess what? It does both. Because our cell not only in periods of starvation requires glucose, it also requires ATP for survival, right? And if we can have aerobic respiration occurring, why not go for that? Because it is producing many more ATPs than the anaerobic respiration. So alright, so pyruvate is a commander, right? Here it is going to make this oxaloacetate go in both directions to secure borders here into the TCA cycle as well as into the phosphoenol pyruvate. So both ends are secured, right? So oxaloacetate goes both ways. It enters the TCA cycle, like I just mentioned, it is an intermediate, and it also converts to phosphoenol pyruvate, which pyruvate itself could not, by an enzyme called phosphoenol pyruvate carboxykinase, or PEPCK, right? That's the enzyme. Now coming to amino acids. Amino acids can give off alpha keto acids, one of them being pyruvate, the other being alpha ketoglutarate. Pyruvate I just explained. Alpha ketoglutarate also converts into oxaloacetate because it is an intermediate of the TCA cycle, and it has the capability to transform into oxaloacetate. So now, okay, so one thing I forgot mentioning is that all the amino acids but two, leucine and lysine, participate in gluconeogenesis. You can remember it from loser because loser starts with an L, and so do lysine and leucine. So they cannot produce glucose from gluconeogenesis. Okay, coming to the conversion of pyruvate to oxaloacetate, and then oxaloacetate to phosphoenol pyruvate, it's not as simple. What happens is that, pyruvate was converted to oxaloacetate, and this reaction is occurring in the mitochondrion of the hepatocytes and the nephrocytes, which are your liver cells and your kidney cells respectively. So this reaction is occurring in the mitochondrion of those cells, right? Remember this. By the enzyme pyruvate carboxylase. Why did we convert pyruvate to oxaloacetate? Because pyruvate could not be converted to phosphoenol pyruvate, but phosphoenol pyruvate can be converted to pyruvate, right? Where at the moment we're not proceeding our steps, we're not going into glycolysis, we are going in the reverse direction to produce glucose. So oxaloacetate tries to get into the cytosol. This is the cytosol. Oxaloacetate tries to get into cytosol, and this is the outer mitochondrial membrane, but it is prevented so because it does not have a transporter to do that, right? So what happens is that alternatively, this oxaloacetate tries another method. Now this oxaloacetate is very clever. It was sent by its commander to go back and yield glucose, right? So what it does is that it disguised itself as malate, as another compound, malate, so that it can now enter the cytosol to cause destruction and conquer the land, right? So it forms malate by an enzyme called mitochondrial malate dehydrogenase, right? Mitochondrial. This malate then enters through this outer mitochondrial membrane into the cytosol of the cell, and once because malate has a transporter, right? And when it enters, it oxidizes back to oxaloacetate. So oxaloacetate, it shows its true self again. And this conversion is done by cytosolic malate dehydrogenase. Alright? Now this oxaloacetate can easily convert to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase, or PEPCK. We learned earlier malate was by the enzyme malate dehydrogenase converted to oxaloacetate. And when there is a dehydrogenase, always remember that an NADH molecule is liberated. That NADH molecule will be helpful in this step. If you remember from glycolysis, that glyceraldehyde 3-phosphate required an NAD positive molecule to form 1,3-BPG and it yielded an NADH molecule. Now to go in the reverse step, we need an NADH molecule available to be converted to NAD positive so that this reverse step can occur to convert 1,3-BPG to glyceraldehyde 3-phosphate. And that NADH is provided by malate dehydrogenase, which converted the malate to oxaloacetate in the cytosol. This is the cytosolic malate dehydrogenase. And then oxaloacetate obviously formed phosphoenol pyruvate by the enzyme PepCK, right? And then all the steps occurred in the reverse order, right? In the reverse order using the same enzymes until they reach fructose 6-phosphate, fructose 1,6-bisphosphate, right? Once they reach fructose 1,6-bisphosphate, if you remember, fructose 6-phosphate converted to fructose 1,6-bisphosphate by the enzyme phosphofructokinase 1. That enzyme considered as a very loyal emperor, right? All the other enzymes are traitors, so they easily got into the trap of oxaloacetate, but phosphofructokinase 1 remained steadfast. It did not, go into that. So we needed an alternate route. That alternate route was provided by another enzyme, which is fructose 1,6-bisphosphatase. All right. I'm sorry, there was an error in my diagram. First of all, what happens is that once fructose 6-phosphate is made, it is converted to glucose 6-phosphate by the enzyme phosphoglucose isomerase, which is the same as in glycolysis, right? Then what happened was that the king, the emperor of the defending state, the defending empire, what he did was that he hid himself because he knew that defeat was near, defeat was close, so he hid himself in an endoplasmic reticulum, right? So the forces of oxaloacetate, which is by the way now converted into fructose 6-phosphate, this oxaloacetate, they wanted to know a local person who would know the route to the king, right? So what happened was that luckily they had a helper in the form of an enzyme known as glucose 6-phosphate translocase, which translocated this newly formed glucose 6-phosphate across the endoplasmic reticular membrane inside the endoplasmic reticulum, where now it can convert to glucose by the enzyme phosphatase. End of their empire, right? They're done. So glucose is formed. Now this gluconeogenesis is, it is heavily stimulated by an excess of ATP. Why? Because if you remember, I explained earlier that oxaloacetate had two paths to go. One was to convert to phosphoenol pyruvate, the other was to go into the TCA cycle, the Krebs cycle, to form ATP later on, right? And it took both the routes because it had to secure both the borders. It was at war, right? Now, once it got the signal that ATP is being synthesized more and more, then what happened was that it drew some of its forces as a backup in this region to increase the gluconeogenesis, the process of gluconeogenesis, right? So more the ATP, more the stimulation for gluconeogenesis to occur. Now comes the regulation of gluconeogenesis, which is very important. There's a hormone known as glucagon, which acts in opposition to insulin. Insulin is secreted when your glucose concentration in the blood is high. Now, gluconeogenesis was initiated in the first place because the glucose concentration in the blood was low, right? So another hormone, glucagon, takes its place. So what glucagon does is that it has three important functions. Number one is that it reduces the concentration of fructose 2,6-bisphosphate, which is an inhibitor for fructose 1,6-bisphosphatase and is a stimulator for phosphofructokinase 1. So thus it is stimulating by lowering the concentration of fructose 2,6-bisphosphate. It is increasing the activity of the phosphatase enzyme, which is what we need, right? Secondly, what glucagon does is that it inhibits or inactivates the hepatic pyruvate kinase. Now, pyruvate kinase was supposed to convert phosphoenol pyruvate to pyruvate, which is not what we need right now. We need to go in the reverse direction. So more and more phosphoenol pyruvate is there to produce glucose, right, in the reverse process. So it shunts it back. Third way how glucagon affects gluconeogenesis is by increasing the transcription. Because it is a hormone, it has that ability to increase the transcription of the gene that is encoding the phosphoenol pyruvate carboxykinase, which catalyzes a reaction of oxaloacetate to phosphoenol pyruvate, thus favoring gluconeogenesis. Now, insulin, what it does is that if there is low insulin, it increases the mobilization of amino acids, right? You might have heard that in people who are suffering with diabetes type 1, who have impaired insulin secretion, even those with type 2, because they have a reduced sensitivity of insulin towards insulin, you'd see them lose weight a lot more than other normal people. Why is that? Because it helps to mobilize the amino acids. It increases the hydrolysis of proteins to form amino acids, and more gluconeogenesis occurs in such patients. Then acetyl-CoA is also a stimulator for gluconeogenesis, and adenosine monophosphate is an inhibitor for gluconeogenesis because ATP is inactivated, right? Okay, so the three enzymes, to sum it up, three enzymes that were the most important in glycolysis, namely the hexokinase, the phosphofructokinase 1, and the pyruvate kinase, they were the most difficult for oxaloacetate to combat, right? Because oxaloacetate had to choose an alternate route. Hence, they are the most powerful enzymes of glycolysis, which cannot be, made to deceive the empire. And that's all for today. I really hope you understood this topic. And if you like the video, please like, share, and subscribe.